I'll use this. So it's going to be a wide field, but not really imaging. So we're just trying to assess a certain volume of tissue quantitatively, as I'm sure that most people in this audience are interested in non-invasive characterization of various lesions and complications. But for that, we also have to understand, first have a good baseline description of the healthy site. So this is what I'm going to be describing today. Okay, let's move on. So the goal is to have a non-invasive and possibly quantitative assessment of chromophore contents in human skin. And the specifics of our work, in contrast with some other approaches that we've seen already this morning, is that we are going to use a combination of optical spectroscopy and a lesser known technique called photothermal radiometry. And the combination of two gives us relatively robust assessment of those specifics. And the analysis is going to be inverse Monte Carlo. So outlook, as said, is characterization of some lesions. After we have assessed the healthy baseline, we can then go and look at some modifications that we see in the structure and the contents. Perhaps a study that's ongoing in our group is analysis of hemodynamics in traumatic bruises for forensic science interest. And maybe also in the future studying and monitoring various laser treatments like removal of vascular lesions, tattoos, or skin rejuvenation. So diffuse reflectance spectroscopy, when I use this term, I mean diffuse reflectance spectroscopy assessed by means of a small integrating sphere. It's small in order to assess various areas on the body. And as you can see in this graph, already a huge difference, not surprisingly, between a healthy site and bruised site. And I just want to comment on some previous attempts at this problem of analyzing the bruises, that fitting diffuse reflectance at each wavelength with the analytical solutions derived within the diffuse approximation theory has serious drawbacks. For one, it cannot account for this so-called red loss artifact, which arises because the light at the longer wavelengths is lost from the integrating sphere because the effective path length becomes comparable to the orifice. And so the one-dimensional theory cannot account for such effects. But perhaps more importantly, diffusion approximation is known to fail close to the skin surface, especially under surface irradiation conditions. So for these two reasons, we are going to apply inverse Monte Carlo approach to avoid those pitfalls. But there is another fundamental issue that we cannot avoid even by doing the Monte Carlo, and this is that DRS is fine as long as you have a homogeneous bulk tissue, right? But in a layered tissue, everything is lumped together. So on one hand, the depth distribution of your chromophore is going to affect the data, but not to the extent that you could actually easily assess that in parallel with their contents. You just don't have the sensitivity. So this is the fundamental limitation that we want to overcome. So some previous study have just used some average data for this depth of the specific layers in skin based on anatomical atlases, but this can be incorrect and inaccurate and lead to some artifacts. So going on to the pulse photothermal radiometry, what this encompasses is you take a single pulse of a light, perhaps from a pulse laser, a short pulse laser, impinge it onto the object, and then as a result of non-homogeneous heating of the object volume, as the heat redistributes inside the tissue, the radiometric emission in the mid-infrared range is going to undergo transient change. So if 0 represents the baseline value of the infrared emission, then we see this peak start, and for example, in healthy skin, we see immediately huge difference from the bruise. So the signals contain information on both the amount and the depth of the absorbing features inside the skin. So here's a little bit of the theory behind it. So on one hand, so if this is the baseline temperature inside the tissue, this is the skin depth, which is uniform, then it becomes disturbed by the laser absorption, and as the heat diffusion takes on, over time, the radiative emission on the surface is going to undergo this transient change, which can be described by, so the temperature field evolution can be described by means of a Green's function approach starting from the initial temperature-induced temperature profile, the laser-induced temperature profile. And then we also have to take it into account that we are not strictly imaging the surface because the wavelengths that we are acquiring actually have a finite penetration depth. So we actually have to take an integral over a certain depth and accordingly attenuate contributions from each layer. And this is because the thicknesses of interest in this problem are actually comparable to the penetration depth of that infrared light. By bringing the two together, we get this integral formulation for the signal, radiometric signal as a function of time, as it depends on the initial laser-induced temperature profile. And of course, it is a very ill-posed inverse problem to try to assess this profile from the radiometric transient. Okay, our group and some others have spent years on this problem, and by introducing several advancements, we have then shown, for example, in vitro studies of some collagen-based phantoms mimicking optical and thermal properties of skin, that we can assess the depth of the included absorbers quite accurately, just based on co-registration with high-resolution MRI in 3D. And we see that this depth of the inclusion, which is absorbing, is really one-on-one with the MRI. So this is like in vitro testing, which gave us some confidence to pursue in vivo work. This is the experimental protocol of the Bruce study. So we went through, I think, about 24 people so far, different ages, both genders, fair skin. And so for the PPTR part, so pulse photothermal radiometry, we use a 1 millisecond green laser, quite low incident dosage, and a fast infrared camera. Typically, we acquire at 1,000 images per second. Setup is pretty basic, and by taking those radiometric transients that I showed you before and applying the inverse analysis to extract the temperature profiles, we can get this very reasonable temperature rise inside the healthy skin, in indicating the epidermal absorption and some residual absorption inside the dermal blood. And then in the bruise, the spilled hemoglobin induces very strong absorption here. So this was now solving the first inverse problem from the measured radiometric transient back to the initial temperature profile. But now we are interested in optical properties in the chromophore, so we should actually solve another inverse problem to answer the question, how did these temperature profiles arise? What is the content of the melanin and content of the blood that gives rise to this exact profile? So this is another inverse problem now on top of the previous one. To ease this second step, we apply a simple geometrical model of the skin. For example, a two-layer model in this case, where we have an unknown thickness of the epidermis and dermis underneath, of course, the melanin content, the blood content, oxygenation, and we have to assume some scattering properties, and we take this from Jacques' recent paper, just a combination of Rayleigh and something me-like, right? And so this is known to most in this audience, so I'm not going to bore you with this. So basically, we have now four unknowns for this model, and the approach that we take now is a model-based fitting, right? We use a multi-layer Monte Carlo approach at the wavelength of irradiation, try to guess some of these parameters as initial guesses, and compute the temperature profile that would result from such a structure under this 532 irradiation. And this is, okay, this takes some time, but it's been parallelized by computation on a graphics board. And we use 10 million photons per run. And from this, we have the forward problem formulation, which is the easy one. So form the profile to the signal, and then we compare the signal with the measured one, and use a nonlinear minimization routine to close the loop. And thereby, we can assess the epidermal thickness, the melanin content, and the blood content that actually corresponds to the measured radiometric transient. Now, the saturation level is here grayed out because this technique only has sensitivity for the absorption coefficient, which obviously will depend on oxygenation and content. So we have to fix one or assume one in order to assess the other one. I'm not going to defend this. This is just the first step in our thought process. So I just wanted to illustrate that we can get here a very nice match with the data. So the black is measurement, the red is best fitting simulation signal. And extracted physiological parameters seem very reasonable, 170 micron epidermis, okay, perhaps a little bit on the thick side. Melanin content 1.4% and blood content 2.7%, and of course oxygenation was assumed. However, okay, these are the corresponding temperature profiles, they look very reasonable. However, it is important to notice that the good fit doesn't guarantee the accuracy of your results. Because the signals are so featureless, there's a huge crosstalks between the three channels, so different combinations of them are going to give very similar signals. So it pays really to look at the correlation metrics, and the off-diagonal elements being very large indicates you have a huge crosstalk problem. So these are not really independent channels as far as the data that you have available are concerned. So, and this is the reason why we have to complement one technique with the other in order to decouple those crosstalks. So as said before, DRS, by using a small integrating sphere, ocean optics, everybody has it pretty much. I just wanted to point out that when you're using these small integrating spheres, there's a not so well-known artifact in this community, which is called the single beam substitution error. And what happens actually is that when you're substituting your white standards, which is used for the calibration of the lamp and such, with your sample, actually the fluence, the light field inside the resonator has changed, but the software assumes it has not. So there's a back influence of the sample onto the conditions of the measurements. You have to remove this artifact in order to have a correct reading of the actual reflectance. And we have studied this problem and showed in a recent paper, 2014, that it's actually not so difficult to remove, but as you can see, the difference is quite substantial and can wreak havoc with the subsequent steps of the inverse analysis if you don't do this correctly. But it's not so difficult to do. It's actually pretty easy. Okay, but now that we have introduced DRS, in addition to PPTR, it turns out that two-layer model is not sufficient anymore. There's no way to fit already the DRS data, diffuse reflectance spectra alone by just considering two layers, because the 600 nanometer wavelengths and beyond go just too deeply, so we have to introduce the adipose also. And here it's known that the introduced scattering of the adipose can vary a lot from sample to sample. So we have introduced like a simple ansatz, which corresponds to the what we believe are most rigorous results from Elena Solomatina in 2006. She had two samples. These two green curves indicate two different samples, and we have just found an ansatz which matches both of them by just varying this amplitude A, and then we allow this A to go even below a little lower and higher respectively. So now we have actually two unknowns for the two thicknesses: the melanin content, blood, oxygenation, adipose, amplitude, and we also allow a little bit of blood into the epidermis because it's going to encompass all the melanin available, so it's going to include a little bit of the papillary blood as well. So the mathematics, the schematics of the mathematical approach are like this now. So in addition to the Monte Carlo in the green to count for the photothermal experiment, we now run 13 Monte Carlos for 13 strategically selected wavelengths, and we have 7 or 8 variables to optimize by now matching both of these data sets to the measurements. And this is doable. Again, a massive parallelization helps. And here you can see the match of the PPTR signal transient as well as the DRS fitted again objectively at the same time to these two data sets, and we get the epidermal thickness that is reasonable. Melanin content is similar to before. Very little blood in the epidermis. Around 2% in dermis. The oxygenation seems rather low though, right? Also the dermal thickness of 0.5 millimeter is not very satisfying, but it's just a start. I could, but nobody else succeeded. There's no clip to begin with.
Oh my gosh, clip. Oh, okay, I'm sorry.
Technical. There's something here. technical break. Alrighty. Okay, so we still weren't totally happy with this fit, and also the results are a little bit controversial, so we have introduced an additional layer to account for the difference between the papillary dermis and the rest of it. And this introduces two new variables, so the parameter space is expanding as we do that, and here are the results. And interestingly enough, the match did not improve at all, so we are kind of stuck at this point. We're quite unhappy with this part here, here, and so the idea was, okay, after some thinking, we said, okay, perhaps we have erroneously assumed that the scattering properties are actually known. Perhaps we should have allowed the scattering also to vary on individual site basis, right? And after doing that, can you see here the difference? Observe the difference here. Here it undershoots and overshoots. After correction, everything falls into place. Also the DRS, this was before, this is after. So the only difference remaining is this band in the green, but this is the band where it's known that the beta-carotene absorption is prominent, and we are not including beta-carotene in our study, just not to complicate things, because they are of no interest for us at this point. And you can see now that we got this very nice match. The blood content is around 2%, and now the oxygenation has raised to 48% in the reticular dermis. We're not still ecstatic about this number, but a lot of other groups are reporting similarly low numbers, but I'm not sure if we really understand how this comes about. The melanin content around 1%. This is type 1, type 2 kind of skin, probably closer to type 2. Anyhow, so the scattering properties now, instead of having this fixed prescribed spectrum, we have allowed it to vary, to adjust itself by this familiar ansatz, and the values of 62 per centimeter at 500 nanometers and 1.8 for the power, scattering power, very reasonable. And as said, it brought down the residual norm for the DRS part by a factor of 5 or so, for the thermal part by a factor of 10 or more, and the total residuum by about a factor of 10. And this is the comparison. So this is the Jacques assumed spectrum, and this is our resulting spectrum. And a correlation with some literature data. These are various spectra that we found in the literature, and this is our result in blue, and it kind of fits. I'm not going to discuss the details. Or perhaps only this. For Solomatina, we can see here the very light is actually epidermis, and the underneath Solomatina is dermis. But see, we only have a single scattering properties prescribed for both epidermis and dermis. It may not be correct, but this is our starting point. So the shallower, the shorter wavelengths don't penetrate as deeply, so the result is biased towards the epidermis value probably, and vice versa here. So I think this explains this discrepancy. So this is actually another person, a woman previous who was a man, and we see exactly the same effect by using fixed scattering properties from literature. Not so satisfying match on the left-hand side, and on the right-hand side after correcting, and which actually the fit introduced the larger A and the larger B as compared to Jacques. We have this very nice fit, and the physiological parameters are all within expected values. Even the oxygenation in reticular dermis goes up to 80%, which is really, really nice. So I'm going to wrap up with this. So basically, we combining this spectroscopic and spatial, say, approach really locks in the parameters of the skin quite robustly. If it's accurate or not, we still have to ascertain. Assessment of scattering properties appears possible, still under further investigation, but we feel that this is a good baseline to approach analysis of cutaneous pathologies and such, as I mentioned initially. Thank you very much.